Biochemistry will not fail you because you are a bad student, but it will fail you if you studied the wrong way. So, in this video, I'll teach you how to approach the subject, how to use your resources properly, write answers that actually examiners will give you marks for, how to make efficient notes, and if you can stick through the video, I'll be adding my own notes as a sample in the end. Hi everyone, this is Apurva. I'm currently a third year MBBS student at GMC Ratlam, and in this video, I'll teach you everything you need to know, starting with the approach. So, biochemistry is all about cellular events. So, these cellular events have an impact. These cellular events are regulated or controlled by certain factors, and if there is any problem in this, there will be a disease, right? So this is what you need to understand. You will be studying in the subject, and almost every unit will be classified into a classification of whatever molecule you're studying and its metabolism. Moving on to resources, I am a big fan of Vasudevan, but if you don't like the book, you can follow any other one. Vasudevan ke alawa, there is Satyanarayan, there is Pankaj Nayak. So what you're going to do, you're going to read the book. You'll first flip through all the pages, look at all the subheadings, all the diagrams, all the flowcharts, and then you'll read it again once you have the importance list, right? Read the entire book and make notes only for the important topics. And try to make sure that you make your own notes. I feel like those are usually better than any pre-made notes. Then we have Jambulkar notes. If you have no time, if you've not really studied well and you're in a pinch of time, these are really nice. They are very shortened. So you have to make sure that you're not skipping out on the topics that are in this notes. Don't make notes out of Jambulkar, right? Do not shorten it any further. And it has great mnemonics. So if you do not have enough time, you can go for Jambulkar. Moving on to the best way to take notes in biochemistry. So for classification, we are going to make a flowchart. So you divide your things into whatever, and for important molecules, we are going to add whatever details we need in that flowchart itself. So you make sure that your flowchart has enough space to occupy the main bullet points for whatever molecule you're studying. Then, as for metabolism, taking urea cycle as an example in this case, first of all, you draw the cycle, the skeleton. Then you will add the enzymes. Once you've added the enzymes, you need to add the energy metabolism, right? So, if there is ATP being made, if there's ATP being used, if there is a production of NADPH, whatever these energy rich molecules are, and then at the very end you will add all the diseases that will happen if there is a problem in the enzyme. And once you've done that, make sure you make this cycle very big, right, so that you have enough space to add all of these things. And at the very end you will add the regulatory factors for this cycle. So in this example, I'll show you. There is this step in which this first enzyme is the rate limiting enzyme, and it is controlled by high diet protein, which causes induction, which causes this enzyme to be formed. Then there is 2 ATP being used in. But if this enzyme is absent or defective or in less quantity, you will have what? You will have hyperammonemia type 1. Now, in the cycle itself, you will add a few bullet points, keywords of how you will know if your question is asking you to diagnose hyperammonemia type 1. You write it in very short. Similarly, if the transporter here, which will transport L-ornithine, is defective, you will have triple H syndrome, and then you'll write a few things about the triple H syndrome. What are the three H's? How do you know? What are the clinical features, right? So this is how you add it. And in this case here, we have arginosuccinate entering another cycle to create an energy-rich molecule. So we will add this as well, right? So in short, in your one flowchart, you need to have all the enzymes, all the diseases and the clinical features, their energy and the regulation, which is inhibitors, poisons, and genetic control if there is any. So this is how you take great notes. If you study like this, you will spend less time flipping through the book, and it will be much more easier for you to fetch marks because you will have this one thing, and in this you'll have everything written. So you're also saving your time at the same time. Now, as for answer writing, first of all, you need to make the flowcharts, you need to make the cycles. Those are very important. And it is okay if you cannot remember all the enzymes, but you need to know the name of the important ones, like the rate limiting ones, the ones which are irreversible, maybe in a reaction where all of them are reversible. So you make sure that you are using underlines or boxes or stars. So when you're writing the exam, make sure you underline those keywords or you make a box around them. Again, similar to anatomy and physiology, this answer also has to be divided into heading and subheadings and further bullet points so that your presentation is good. And these are usually the headings that you will be needing, which is classification, metabolism, regulation, energy tally, and disorders or enzyme abnormality. Moving on to strategy, first of all is your lazy man strategy, because I get it. Biochemistry is not something students are very hyped up about from the beginning of the year. So you focus on anat and physiology majorly. Cover the syllabus a few days before the test. You can prefer Jambulka for this. And then once you have somewhat of an idea about what is going on, by the time your second sessionals or maybe your pre-university rolls in, start studying the book again. So now you already know what are important, because Jambulkar is a list of importance indirectly. You read those from the book, you make sure you have everything down, and a lot of these cycles are very rote heavy. I mean, there are a lot of mnemonics, but your main aim should be that you're able to recall. So make sure you revise your mnemonics, make sure you write and practice your flowcharts. Then we have the summer and strategy. The students who study a little bit throughout the year and then score well. So keep up with the class and or the exam schedule. Make sure your syllabus is finished, whatever is coming in exam. Ensure there is not a lot of backlog, and start reading the book from the beginning. As your professor is teaching you, go back to your hostel or your room, whatever. Read the book, read that topic, and pay special attention to the important topics because those usually show up in your exams. Make sure you practice drawing the flowcharts again, the same as the lazy man strategy, and you have to be consistent. Lastly, closing notes. My only advice is follow only one book. You do not need a lot of books for biochemistry. All of them more or less have the same content. Don't Please don't entirely just skip the subject and be blank in your next year, because there's a lot of pathways which will again show up in pathology and medicine again, right? You do not want to be very clueless about those. Make sure you remember the clinical correlations to the topic very well. If there is any applied, if there's any disease which is occurring, if there is any therapy which is being mentioned, make sure you know those, because those are high yield and they will again come back in the subsequent years. And yeah, repeat, recall, and rinse. It is a lot of memorization. You'll have to repeat or else you'll lose it. These are my notes for heme synthesis that I took in my first year. So, see, I start the topic with heme synthesis. Where is it happening? Cells that will slowly become RBCs. And then I've mentioned which enzyme. And then I have written that ALAS1 is inducible, and in liver also heme synthesis is happening. ALAS2 is not inducible. Location I've written as mitochondria and cytosol. In both the cases, it is happening. Some great doctors palpate heart undercover. This is the mnemonic, you'll find it everywhere. And over here in pink, you'll see cytosol, and there is a line under which there is mitochondria. So these two things are happening above in the cytosol, and then it enters the mitochondria. Then again, the steps are going on. I have written ALS synthase. Repression is caused by lead, heme, hematin, glucose. If these substances are there, ALS synthase will be less. And plus barbiturate, which means if the person is on barbiturates, ALS synthase activity will increase. Now in green, I have circled it and written pyridoxine responsive sideroblastic anemia. If there is any problem in this enzyme, this is the disease that might happen, right? It is the same in all of these steps. And again here, again after corpoporphyrogen 3, it enters mitochondria again. So in this one flowchart, I have everything the question might ask me in one go. Oh, by the way, again, there is also negative feedback by heme, which will inhibit ALS synthase. So everything is written in one big flowchart. Then I've written porphyria separately so that it's easier for me to understand, because there was not a lot of space on that page. So see, I've written acute intermittent porphyria, so abdominal pain, vomiting, neuropsychiatric manifestations will be there, right? So if there is a question which asks me to diagnose a porphyria, I already know what in each porphyria I will be seeing. So, for example, in cutaneous tarda, there is neurovisceral photosensitivity. It is neurovisceral. There is photosensitivity. So, I will be able to write my answers much easier. The keyword in erythropoietic porphyria is that it is recessive and it has potvine urine. In the book, there is some therapy or treatment given for them, that also I have included here, that you can give them glucose infusion, you ask them to wear sunscreen, you give them vitamin A, C and E, hematin infusion kar sakte hain, and all that. And again at the bottom of the page I have a mnemonic. So this is the most efficient way you can go about biochemistry, and I hope it helped. Best of luck.